So, good morning, everybody. I hope you will understand me, and I will give now a little bit complicated lecture, a very overall lecture about many things, what we're thinking. But the special theme I have is recognizing and learning from nature. And this is a very complex theme, how, in which way we are recognizing nature and what we are learning from nature. So, first we have to think, what is observation? When we are observing some things and then we are recognizing it. And this is a formal way for recognizing these organisms and organism groups according to a theory, and this is classification theory. And in this theory, we have put two basic definitions. We have to define the objects. The objects are defined by possessing a single character at least. If there is no character, then it is not an object. So an object must not be, should not be a real things. You can feel, but it could be a word or a sentence or such things. And then now by the second definition, what's a character? And a character in simple form, it's an information unit. So classification, sensu stricto, is the grouping of objects by their characters into classes, and these classes has to possess similar characters, and to differentiate from other classes, they are different characters. This is classification, so the grouping. And the next is, we have classification in the other sense. This is meant by identification, which is the assignment of an object to a class possessing similar character states. So a preformed class system, and then we put an object according to their characters to this class. And this is identification. So recognition is the assignment of characters to an object and leading to identification. So characters in classification sensu stricto, we have two types of characters. We have the basic characters, they are fixed character states, determining the objects. So for example, here, the members of this Congress. This is a basic character. And we have the classificatory characters. They differ in the character states between the objects. So for example, in this group here, we have males, we have females. According to age, we can classify, according to body weight, according to height, and so on. So, essentially, a grouping of objects into classes is based on the relations between the objects. And the objects are defined by the basic characters. And then, this grouping uses the classificatory characters. So, the position of objects in an hierarchical classification is also important for nature. We have first-order objects. They are all characterized by character set 1. Second-order objects are first-order objects characterized by character set 2, and so on, third-order objects, or in formal way, you can see here the formulation. And this, we have first-order objects in biology are organisms. Here. So, first-order objects are organisms. And the basic character for an organism is life. And life is determined by a set of characters. So, organization, growth, development, response to stimuli, reproduction, homeostasis, metabolism, and so on. It is very difficult, not all should be here performed. But, very important, organisms are individuals, being spatio-temporal units. And when hard-coded changes occur in time, then this is development. Second order objects in evolutionary biology are species. But the basic character set for a species is not yet determined, leading to different species concepts. I will speak later about these concepts. So we have explorative concepts, we have operational concepts, and we have family resemblance concepts. But also species are individuals because they are spatio-temporal units. And when changes occur in time, this is evolution. So, I will show only shortly the organization level of the biological world in the biosphere, looking for individuals. And we have here, as individuals, the multicellular level and the population level. Look, it is not a species level, it's a population level. And additionally, within some single-celled forms, also these single-celled organisms are individuals. So, recognizing individuals by humans without an optical support, you can see here the complete system of organisms, bacteria, archaea, and eukaryota. And within systems, you have only the slime molds, the animals, the fungi, and the plants you can see detect by eye. For all the other forms, you need optical instruments. Now, we can go to the eukaryota in detail. And you can see here the phylogenetic relationships between the eukaryota, the today classification. So we have the SAR group, we have the SAR group, the Archaeplastida, the excavata, the Amoebozoa, the Opisthokonta. And within this group, we can have larger organisms you can detect by eye directly. And you can see we have here the Straminopiles, the brown algae, the rhodophyceans, the Chloroplastida. These are plants. Within the Amoebozoa, you have the Dictyostelia, and within the Opisthokonta, you have the fungi and the Metazoa. So the fungi are often treated as plants, but they are much closer related to ourselves. So, only to give a small picture about this group. So you have the SAR group, the Straminopiles, and you can see here brown algae, Sargassum, and here the kelps, around about 80 meters in height in the ocean. Archaeplastida, the red algae in the ocean. Chlorophyta, these are the green plants. We have the charophyta, the mosses, the ferns, the horsetails, the gymnosperms, and the angiosperms. These are the largest group. And next, we see the other group with the Amoebozoa. Here you can see the Dictyostelia, the slime molds, and the Opisthokonta with two groups, the fungi, cup fungi, gill fungi, and the Metazoa with sponges, jellyfishes, scallops, snails. So these are Protostomia here, this Protostomia, and these are Deuterostomia here. So it's the position of the original mouse and annel. So, next step, we have the relation between the subject, the human now, and the object, the organism, in biology. So, subject and object, we have recognition. But from the object, we have also the response to the subject. And further, we have, from the subject to other subjects, we have the second order response, and this is called learning. We have a direct response and an indirect response. So when they are regarding the effects of the object to the subject, then this is an indirect learning, and a direct learning when the subject says to the other subjects, gives direct the response, showing the response. So, for example, Relations between subject and object. So we have here a fungi, a cup fungi, and we know by experience this is a parasol mushroom, and we have a very similar form, but this is identified as a parasol mushroom, and this is extremely tasty. So it affects the parasol mushroom community because the people are collecting them, and so reducing the population. Then, on the other way, we have now this fungi, and this is, by the name, as you can see here, this is a death cup. And the response of the death cup is, it is really totally lethal. So, the effect of this relation is that this population will not be reduced as the parasol, but possibly this population will be shrinked in some way. So, next, reasons for recognizing organisms and learning from organisms in ancient societies, and we have the hunters and gatherers, and also some animals, and the recognition of huntable wildlife, recognition of eatable plants and fruits, and recognition of medical plants, and they have to be aware of predators. We have also ancient societies, so we have stock breeders, so detecting animals for breeding in herds or in stables, recognition of predators for the breeded animals, and we have the farmers detecting plants for cultivating in fields or in gardens, and the recognition of organisms like fungi, animals, and other things that is destroying the cultivated plants. And this leads to reasons recognizing organisms in modern societies. You can see here many facts important for molecular genetics, pharmacognosy, ecology, biodiversity, biogeography, evolution, phylogeny, conservation biology, and so on and so on. Many facts, many important facts. So, the relations between humans and organisms leading to evolutionary theory. So you have here the subject, and here objects in the classificatory sense. So, and the relation is, they are extremely similar, extreme similarity, and the response is, there must be a strong genealogical relationship. And this is the idea, the basic idea of evolution. So, the higher order of objects we find now is the nth order objects characterized by a character set n, and these are groupings of organisms in a hierarchical system named categories. And the history of classifying the organism world into higher orders, the first one was Aristotle, and he has written books about history and the generations and so on, and the grouping traits are with and without blood, the number of legs, and the reactions. Rational, like humans, sensitive, or vegetative, like the plants. So the next really important fact is the introduction of the Systema Naturae, the natural system by Carl Linnaeus, Carl of Linnaeus. And he, in his 10th edition of Systema Naturae, he established many things. First, he established the introduction of the binomial nomenclature, so that everybody could understand what there is a meaning of these organisms. For example, this one here, and Linnaeus said, of course, this is the genus name is Felis, and the species name is Leo, and this leads now to Felis Leo, and here is the original page. And you can see here Leo, F cauda elongata corpora el vulvo, and we have the Felis, the genus Felis. Dentes, the premoles, the former are equal. The molares, the back teeth, are the 3 molares, and the ungues are retractile. You can retract it. You have here a cauda. The tail has a bush at the end, and it has a mane, thorace, jubato, and so on. So very simply described. And also, the second matter was that he was introducing the hierarchical system of organisms. And this system, in Linné's sense, started with the variation, the species, the genus, the order, the classes, and the regnum. But then, in the kingdom, the today hierarchical order system is starting with the species, then the genus, then the familia, then the order, then the classes, then phylum for animals, divisio for plants, then the regnum and the dominion. So this evolutionary system of organisms was evaluated, not introduced but evaluated by Ernst Mayr, and in his work, Systematics and Origin of Species and Principles of Systematic Zoology. And he is the main representative of the synthetic theory of evolution. And he said that the extension of the categories by creating super and subcategories allows this representation of the evolutionary tendencies. And this is very difficult and could not be really correct. As I will show in the later example, here we have the position of the lion now in the modern evolutionary system. Starting with the dominion, eukaryota. Okay, this is okay. Next one is the regnum. But here you can see regnum animalia, of course, but we have to establish a superregnum, Eumetazoa. And again a subdivision, but not subregnum, yeah? It is Bilateria. The next category is the phylum, but you have to see we must erect a superphylum, Incedecetes, Craniata, and a subphylum, the vertebrata, the vertebrates. And again, we have the classes category is terrible. Yeah? As you can see here, there are so many Incedecetes. It is impossible to give an evolutionary tendency with the class system. It becomes better with the next. This is the order. So we have the order Carnivora and the suborder the Feliformia. And the family, we have the Felidae. And we have then the subfamily Pantherine. And finally, we have the genus Panthera and the species Leo. And today, his name is Panthera leo, no more Felis Leo. So you can see it's completely, it's impossible to create an evolutionary tendency or phylogenetic tree or the evolution with this system. So it was Willy Hennig, working on an entomologist, working on insects, and he introduced the phylogenetic system very early in 1950, but he was really alone. But in 1966, with the Phylogenetic Systematics, the translation of this book into English, then it really changed, and today it is the basic system we can find. So here, species is a basic unit. It was based primarily on morphological characters. Today it is based primarily on molecular genetic characters. And the classification criteria in his time by morphological character are apomorphic characters, so this is a derived character state, and plesiomorphic characters, it is a preserved character state. So, this is not a nested hierarchical system, because no categories are possible in his system. So, you can see the requirement in phylogenetic system is, in his system, is the bifurcate speciation, starting with a mother species, and we find, according to the apomorphic characters, we get 2 daughter species. So thus we have different types of hierarchies. We have the so-called inclusive, the nested hierarchies. So the objects are grouped into classes in order of decreasing connectivity, and this is based on symmetric relations. So A is in the same relation to B as B is in the same relation to A. So for example, similarity is here very important. Then we have the exclusive, the ordered hierarchy. The connection points within the hierarchy are objects. And these are based on asymmetric relations. So A is in relation to B, but B is not in relation to A. Like the father-son relation, yeah? This is a typical asymmetric relation. So, and now we have the 2 types of hierarchies. We have here the Hoheneggian system. You can see here, these are the living, the extant forms. These are the extinct forms. So parents, and also these are parents. And we have here the exclusive hierarchy. We cannot give here a real order of groups, but in the inclusive nested hierarchy, demonstrated as dendrograms or cladograms, we have here the order of cousins. And as you can see here, we have the first order, these are the sisters, and then these are the sisters, and then we have the first order hierarchy, the second order hierarchy, the cousins, the third order hierarchy, and the fourth order hierarchy. And you can see here these and these 2 are really, they are connected by their parents, so they have a first order hierarchy. And these are connected by these parents, and this group, and these are second order cousins. So within this is the Linnaean system. We have an order, and we can use it. It also shows the phylogenetic relations, but without the real ancestors. So these are only the living forms, and in which grade they are related. So in both systems, species are the classificatory objects. So species now as evolutionary units, you have here to identify and give name, basic characters must be determined for the species, preventing a mixture of apples and oranges. So, basic characters for the species that are universally accepted could yet not be found. This is exemplified by different running species concepts. And we have, as I have shown before, the explorative species concepts that tries to explain how species are created, but most of them lack basic characters, making the species, all species, comparable. So you can see here the different species concepts, explorative concepts. I cannot explain it in detail, that it will last for some hours. But we have the other concept, and these are, and these are the operational species concepts. They do not explain species, but they make them detectable. Delimitations of the phylogenetic species according to the dendrograms, genealogical species, diagnosable species, evolutionary species, phenetic species, the morphospecies, and genotypic cluster species. So many different types, but make them identical, some in some ways. And then we have this family resemblance concept, a very new concept. So it means the species is not a real class, it is a group of classes, and it depends on the family resemblance concept by Wittgenstein. He was explaining, if we try to explain a basic character for games, game is clear, it is impossible, because game, football, golf, poker, these are all games, we understood as games, but we cannot find a universal concept describing them. So we have in these games, some of them are molecular genetic distance, reproductive isolation, ecological or functional differentiation, and morphological phonetic differentiation. So, now I will show you the relation between the genes, the genotype, and the traits, the phenotype, and you know it very well, I think. We have the genomics, we have the DNA, and the transition to RNA, and the transition to proteins, and then the transition to metabolites. But also, the way back is possible, but not a direct way, not always directly, but we have from metabolites to proteomics to transcriptomics and to the genomics. So, genetic changes can be detected morphologically in populations, or not also not only morphologically. Populations are spatio-temporary, limited, interbreeding communities. But we have some things that are clones that are not interbreeding, but we have also clones as communities, asexual reproduction. The earliest of population with a homogeneous distribution of phenotypes is caused by constant allele frequencies and with the dominance of the heterozygotes. So we have the consistency of alleles. We have two alleles, and we have the homozygotes, only allele small a, and the homozygotes too, all only have allele large a, but then we have the heterozygotes, the mixture, and normally you can find the proportions of these types, and you can see here it belongs to a more or less normal distribution. And what's very important, this genotype composition through time remains, of course, stable when it is following the Hardy-Weinberg equilibrium. It is the constancy of allele frequencies. So, for example, this type here, you can see in the evolution through time, it keeps totally constant. But the Hardy-Weinberg equilibrium, the proportions of alleles keep constant in succeeding generations only if you have sexual reproduction and you have large populations, and very important, you have panmixy. The same probabilities of reproduction between all genotypes, and this is mainly also in very large population, it is totally impossible, panmixy. So, for example, the mating between a male and female citizen of Kyoto is much more probable than the mating between a Croatian male and a female citizen of Kyoto and vice versa, which is extremely improbable. So, panmixy also in very large populations, it is not given. So the Hardy-Weinberg equilibrium will be disturbed in any way by mutation. This means the changes and the introduction of new alleles. Then the genetic drift by small populations. Then the migration between small populations. Here only the immigrants count. And last not least, the selection, the preference of the fittest in reproduction. And these three are non-directed random changes, And only the selection, it leads to directed changes. So, only for the non-biologists mutation, you can see you have in your RNA, you have the codons for designing the amino acids, and you can see here the adenine, uracil adenine. Here it is responsible for isoleucine, but when there are changes, uracil, cytosine, then it is not affected. This is isoleucine again, but here only when here on this place is guanine, then methionine, here threonine, here leucine, here arginine, here leucine, and also valine. So you can see this punctal mutation has a very strong effect. Genetic drift is very easily to understand, so they are dying out, we have a so-called bottleneck effect, and only these phenotypes survive, or genotypes are surviving. Migration, only for the population do. The immigrants are counting because they change the genotypes here. The immigrants does not disturb here. And then selection. So we have, normally we have homogeneous and inhomogeneous distribution of phenotypes inducing selective trends. You can see here we have the, we have, on the left side we have the tendency that this homozygote here, this genotype is much more pronounced, and this can lead to transformation. This is called species transformation or anagenesis. And in the other fact, we have the disruptive selection. So here in this case, the homozygotes are preferred, and this leads finally to the cladogenesis, to the speciation event. So that two different species are arising. But the introduction of new genotypes by mutation, leading to continuous changes in phenotypes here, as it is shown here, it must be that then we have the selection, but it could be also not only a very small, it could be, So you can see here, the anagenesis, the species transformation. You can see it from this starting and changing to this form here. Or the disruption of genotype composition. So again, the homozygotes are preferred by, introduced with a new mutation, but on both sides now. And you have here, you see here the species A, then we have the gray zone of evolution, and then the development of two different species. Mostly you can find it here caused by a barrier, mostly by a geographic barrier. So, the introduction by new genotypes can also lead to abrupt changes, as we can see here. And this leads to different explanations of evolutionary trends. And now I will start the speciation models. We have here the split of speciation. So we have two types. We have the so-called gradualistic speciation. This is the adaptive zone is changing, and also with this adaptive zone changes also the species. But then appears a second adaptive zone, and on the border there is a second species evolving in a different way, but also gradually evolving. And also this adaptive zone is changing. In the other case, you have the punctuated equilibrium model. So these zones are stable, the adaptive zones are stable. The second zone is also stable. And then from one zone to the other, very rapidly, there can be border population can cross this barrier and evolve to a second species. And this can turn back to a next species here, and so we have a species C here, yeah, deriving not the direct derivative of species A, then here a species C. So I will explain it now. I hope to can explain it by evolutionary models by planktonic and benthic foraminifera. And here we have, sorry, I have to put here. So you can see here this single-celled individual, but highly complex individual moving. You can see it's very large for a single-celled organism. It is 2 millimeters in size, and it is here walking around this part here. Yeah, it's pseudopods and going on. So these are the richest organisms, single-celled organisms in the oceans and in the marine environments. We have benthic forms living on the seafloor or in the seafloor, and planktonic forms, they are living in the water column. So, what's the effect here? We have a side, and they have a very long history. They are starting in the Devonian, so it means around about 400 million years before, so in the Paleozoic. But in the Mesozoic, and especially in the late Mesozoic, so in the Cretaceous and in the Cenozoic, they are becoming very important. They show all very high evolutionary tendencies, and so they can be used as markers for time, but also showing the evolutionary tendencies. And I will show it. It was a scientific drill in the type locality, the Bardenian, and here it is a group. This type locality is not positioned in Siberia, as you may imagine by our clothes here, but it is in Austria, but it was frozen. It was in February. And you can see here, this is the drill for investigating, and you get the samples, the drilled samples, and these are marls, very fine sediment, and they are very, very rich in these microorganisms. So why they are so rich? Let's have a look to the next picture. And you can see this is today's ocean floor at 2,000 meter depths in the tropics and subtropics. And you can see no terrigenous material. These are only planktonic foraminifera. The total floor is covered by these protozoans. You can see here the size, 500 micrometers, very small forms. And yet then you will find, when you investigate some part, for example, 5 centimeters section of the sediment, as you have shown before, then in sediments of 5 centimeters thickness, with a sedimentation rate of 0.1 millimeter per year, this is very common in 200 meter depths, you get in this 5 centimeters 1,000 generations of benthics. So these tendencies, this variation within the sample does not show the population variations, it shows the averaged variation of the species. Over 1,000 generations in benthics, but here we have planktonics with a much lower sedimentation rate, and this is now 0.01 millimeters per year in 1,000 meter depths. This 5 centimeters of sediment is a result of 60,000 generations of planktonic foraminifera. So this is a really good averaging, showing really the trend, the evolutionary trend, and not the population dynamic trend. So, for example, for both two samples, you can see here now a gradualistic speciation, as I have shown before, with the plankton foraminifera Phocella phocis. This is one here. You can see the age from 14 million years up to 12.8 million years. And here in the eigenvalue, it's a shape constant, but here in the size, you can see here the one trend, as I have shown before, and then the other trend. So, all these variations here, you can see, is the result here, the result of around about 60,000 generations. But in this averaged generations, you can see the clear tendency. Then, next one, we have punctuated gradualism, also in the planktonic foraminifera. You can see here the first trend, stable, then a very rapid transition, gradual transition, and then stability. So this is caused by climatic effects between the Miocene and the Pliocene here, 6 and 5 million years ago. We have also effect for punctuated equilibrium. This is now a benthic foraminifera living on the seafloor. And in the upper part, this is here a single drill. You can see here one first tendency, and then replaced by this group here, and then replaced, suddenly replaced by that group, and finally replaced by this group here. Then we have also other cases of split-off speciations. So we have the gradual quantum speciation. So here we have first a changing form. It is quite distinct from equilibrium, punctuated equilibrium changing. But then we have a sudden change here, and a second adaptive change, transferring, and again we have then the gradualistic tendency. When we have such a continuous, we have not a sharp, distinct event. When it is first, it is stable and then continuous, then it is not as the limit, not a species limit. These are sublimits, because you cannot, you have to put here an artificial limit between this form, between the population here and the population here. They are very, very similar. So you have to look for the whole tendencies to give them a name, a species name. So you can, for this gradual quantum speciation, again, planktonic foraminifera, you can see also, we have seen before, we have here the tendencies, then the sharp distinction, and then again the gradual evolution here. You may say, okay, this is not so really good indicative, but with the angle, the mean angle here, you can see there's a very strong shift. They have the angle here, and up they are totally rounded. They don't have an angle. So, and this is caused, this tendency is caused in any way by the climatic change in the Cenozoic, and you are here with this 5 million years, you can see it's a very strong climatic change we can find here. And the continuous change of the cone-formed shells, as I've shown you before, into groups of Contuso-troncana, it is again in the Cretaceous time, in the Mesozoic, in the dinosaur time. You can see first stability in the Coniacian, and then in the Maastrichtian, it becomes colder and colder, and so you have the tendency. Here, the climate is constant. Here, the climate is changing, as you can see here, by stable oxygen isotope measurements. So, this has been the split on speciation. You have the split up speciations. First, I've shown you before the bifurcate speciation. It is very rare. Normally, you can find, especially in terrestrial organisms or in very shallow organisms, the so-called reticulate speciation. So we have a species A, adaptive zone, then it changes, and it is divided in different subzones, the gray zones, with different subpopulations, and they can copulate. And also we can see here we have the adaptive zone, we have the species C, and here we have at least under stable, more or less stable conditions, then result are 3 very distinct species. Here we have often some form of hybridization, and in our population model you can see here the overlapping of 2 subspecies, but in this overlapping you can take the hybridization, and this can become a special single line. So we find this reticulate speciations, we find this in the evolution of Acropora. Yeah, Acropora corals. So in the Pliocene, you have such a distribution here, as you can see here, because in the Pliocene the sea level was very high. Then in the Pleistocene, the sea level is lowering down, there are islands, so the connection between the different groups was not given, and therefore we have different subspecies, as you can see here. And then in the Holocene, as today, we have 2 groups, 2 survival species, one much more restricted in the Indian Ocean and the other much more restricted to the Pacific Ocean and an overlapping region, especially in the Philippines and in the Indonesian region. So, the gray zone of reticulate evolution in humans leads to racial ideology, as you can see here in the last century, or in the beginning of the last century. So you have the Indians, you have the Australids, you have the Caucasids, the Negrids, and the Mongolids. Reticulate speciation you will find in the fossil record also. As you can see here, you have here the stem group, and then here in this zone you have the gray zone, and then we have here 3 distinct species with the same gradual tendencies. So we will go now to the non-genetic processes influencing evolution. And these are the epigenetic mechanisms. There are 2 groups, the methyl group, the DNA methylization, the bounding, tagging different codons to the DNA, or we have here the histone modification, as you can see here, by the epigenetic factor, and losing the compound form and expressing the gene activity in this region. We can see the epigenetic factors from the genome. We have here the fertilization, they have the fertilized egg, the genome, but then you have 2 tendencies. The gonad cells, they are no longer epigenetic influences, but we have the epigenetic fixation here in these cells, in all somatic cells, in the nerve cell, in bone cell, in muscle cell, in the liver cell, intestine cell, and this depends mainly also on developmental, on the time of the beginning of these effects fixations here. This is a very strong developmental problem, not so much in the gonad cells. We have also environmental influences, so the niche of abiotic factors, you can see we have a gradient, like salinity or temperature and so on. You have, along the gradient, you have an optimal conditions, the range, and we have here the range of good conditions, and then we have the physiological conditions, and the intolerance conditions. And as we can see here, we have the deviations from the normal distribution due to the Weber-Fechner law, as I've explained yesterday, not linearity. So the reaction of the diatoms of the plants in photosynthesis to irradiation measured in micro-Einsteins is of such a form, non-normal form. It is non-linear form. But if you transform it by logarithm of irradiation, then you can get the normal form, the linearization. So,
Next one, you can get the influence of gradient distribution due to biotic factors. You have the deviation due to competition by the same species group. And we have the competition by different species of genus. You can see here how they disturb the distribution form along the gradient. And we have the influence of the trophic positions, of the predator-prey position. You can see here the trophic hierarchy in terrestrial food chain, in the marine food chain. We have the network situation, so many forms are feeding from the same forms. And so they are also in competition, as you can see here. And one of the very well-known form of predator-prey relationship is the Arctic fox versus the snowshoe hare. Even the snowshoe hare is increasing, and then when it's increasing, the population of the fox is increasing, but then it is decreasing, and then also the population of the fox has to decrease, time dependent, time deleted. So, as a finish, we have the evolution reconsidered by the extended evolutionary synthesis in contrast to the standard evolutionary theory. In standard evolutionary theory, I have shown you new variation we find through random genetic mutation. We have positive mutations and negative mutations. We have the inheritance through DNA, and we have the adaptation to pre-existing environments through selection. And now we have the extended evolutionary synthesis. Organisms are not simply programmed by genes, especially in development. There are some regular genes, but not simply programmed. So they are constructed during development. So we have a developmental bias in this form. Environment directly shapes the organism traits. So we have the plasticity of forms caused by the environment, possibly by epigenetic factors. Then we have the co-construction and co-evolution with the environment, and this changes the structure of the ecosystem. So we have niche construction. And last, I hope but not least, transmitting more than genes across generations. So we have to find the extra genetic inheritance. So at this point, I will finish my talk. I hope you will get enough information about how we are recognizing, how we are ordering, and which way we are learning from the nature. Thank you very much.